Hello, this is a presentation on what to expect in Biotechnology 2. The hour there is not correct, that should be 7th hour, but this is Peter Kritsch. Biotechnology 2 is primarily 11th and 12th graders. We do have one 10th grader in there this year, but the previous 2 semesters that I've taught this class, it's been pretty much all juniors and seniors. The overall goal and purpose and priorities of Biotech 2. The goal is to be able to continue to introduce students to biotechnology. The Biotech 2 class arose out of a couple different things, one of which was that I kept finding that I was rotating into the Biotech 1 class a variety of topics, some new ones in some semesters, some ones that I wanted to replace with something else, but they weren't bad topics to get into, and I just had more than one semester's worth for sure of material for the biotech class. In addition, we had students who had wanted to be part of the Biotech Youth Apprenticeship Program, but it just did not fit with their schedule. So this was an opportunity to introduce students to additional topics in biotech, but also give them some practical experience. And so students in the Biotech 2 class are involved in long-term projects where, unlike Biotech 1, the students are completely responsible for the experimental design, setting up the labs, analyzing the results, and communicating the findings. So it's not like students in Biotech 1 aren't doing some of those things, but there's a lot of background lab prep that goes on for that class. In Biotech 2, the students are completely responsible for that. Where we've been so far, students have been engaged in 3 separate projects. The first project that they worked on in the beginning of the semester came about as a result of a discussion that I had with some teachers in a course that I teach during the summer. And long story short, there are multiple stains that could be used to look at DNA in agarose gel electrophoresis. And I used one, and another teacher used a separate one. And basically, we wanted to find out which one works better. Well, that was my question back to the students in Biotech 2. Which one is better? How do you know? And what would you recommend I use in future experiments? And so students designed a project to take a look at that, and then communicated their results in a technical bulletin that I can now provide to next year's group of teachers that take this class. Project number 2, students were taking a look at protein purification. As part of that course that I teach, I was working with a professor from UW who wondered why we were using one particular protein purification method when this other one worked a heck of a lot better. Long story short is I didn't have access to some of those supplies, but he was more than willing to provide some of those supplies to us. And so this came as a question then right back to the biotech students. How do you purify proteins? Which process yields more protein? So how do we measure how much protein is there in the first place? Which one leads to pure protein? Just because you get protein out doesn't mean it's just the protein that you're after. So how do you know? Which one of those experiments do you recommend I use in the future? So project number 2, which they ended up communicating through a lab write-up, then sent right back to that professor at UW. And project number 3 involves taking a look at sequencing DNA. Once you know the sequence, how do you use a computer process called bioinformatics? So using all the different search engines that are out there, the analysis software that's available to determine the source of that DNA. Where we're going next, there are a couple shorter projects that we're going to be working on connected to genetic identification. And then from there, students will be designing and working on a research project that is completely up to them. I'll have a variety of different suggestions for them, but at that point, it's really kind of what they want to do. So it could be a large group project that they decide to do. This particular semester, there's only 6 of the students that are in there, while next semester I've got 21. Or they could choose to work in 2 to 3 small groups. Really some interesting work that's been done as a result of this project in previous semesters. And actually some of those projects are ones that I will be suggesting right back to the students as ones that they could continue to work on. What you're going to see today, though, is really kind of up to the students. I'm not going to take the lead on this one at all. I'll introduce them to you, but my goal is that they're going to be presenting their work. What have they been doing? I'm not going to have them do all three. I think that's going to get a little chaotic, and I'll kind of bring this back to them to have them brainstorm whether presenting one or two of those projects would be better. And then also to save some time so they can give you an overview of how the class works. Um, don't hesitate to ask them about the assessment for the class. In the end, there's no tests that we're doing. It's all based on these lab write-ups and observing how well they're doing as scientists. Every student starts to develop different specialties within these projects. And so while they're all responsible for understanding the details of the projects, which they do communicate through those research reports, in the end, they gain different insights into biotechnology and how science is done. And so, yeah, ask them about how this class works and whether they like this approach or not. If you have questions, looking forward to it, looking forward to your observations, and I'm looking forward to this particular presentation to see what the students come up with.